So welcome to part 2 of chapter 8. We're going to dive into understanding how mitosis leads to exact copies or duplicate cells. And just a heads up here, we're looking at, this is the parent cell, and this cell has now gone, this little arrow right here shows us going through interphase. So notice that the only thing that's different between this cell and this cell, both of which are parent cells, is that the DNA here is duplicated. It's kind of like attached at the centromere, and that's basically the main important thing that's getting ready for mitosis. We still have cell growth. We still have other things going on, but that's the big deal. Is we've got those new parts of the chromosomes formed, and I'm going to throw in the term. You'll see it on the next slide, but I'm going to throw it in now, and that is that these replicated chromosomes, each half of it is called a sister chromatid. Again, I'm just throwing that in now because I'm probably going to use it as I go through this process, this overview process with you. So just heads up. Okay, so that's interphase, and remember that interphase was the majority of the cell cycle, and that the mitotic phase includes mitosis, which is what happens to the nucleus, and cytokinesis, which is what happens to the cytoplasm. So in the first stage of mitosis, we've got a lot of stuff going on, but highlights here, the chromosomes go from this long squiggly chromatin into this chromosome shape. Remember how I said the word chromosome is a confusing word because it's used in many different ways, and here I am using it in many different ways. I'm using it in two different ways in this picture. I'm using it as the shape, squiggly versus condensed, and the squiggly shape is chromatin and the condensed shape is chromosome. But also I'm using it in the term of these are chromosomes because they are individual pieces of DNA and they have, information on them. So there's nothing I can do about that term. It kind of is what it is. I'll often try to say chromosome shape, meaning it's the condensed shape. And here you can really see how those duplicate chromosomes. It's not as clear here, but you can see here how they're attached at that centromere. So, and then there's other stuff going on that I'll describe to you later when we get into those individual steps. The next step is where they line up in the middle. And notice how these little fibers are coming out and attaching to those chromosomes. Those are called the spindle fibers. And those spindle fibers are attached to these little central areas here, and those little central things are called centrioles. Notice how the centrioles start out in the the cytoplasm, and they start to go to the far sides of the cells until they're eventually in the second stage on opposite sides of the cells. In the third stage, we have the spindle fibers shortening up and the chromosomes separating from each other. And then in the fourth stage, we have the nucleus reforming. You'll also see that we have this pinching in happening. Your book talks about the pinching in as part of the fourth stage, but it also talks about it as being separating the cytoplasm. So it's a little confusing the way your book presents it, that's all. So again, we have to get ready for mitosis, and we get ready for mitosis during interphase. And the most important part of getting ready for cell division is chromosome replication. And again, remember how I showed you earlier, I'm going to flip back to that, where we talked about DNA replication right here, yeah? And I talked about how these two things that look separate from each other, these two identical daughter DNA molecules, are actually attached at the centromere. So if we look ahead to this, when we cartoonize it, we can say, oh, here's one chromosome, and then we have the identical copy being made attached at the centromere. And there's your word centromere. And it's in this X shape. Attached to the centromere, and each half of this chromosome, and this is one chromosome right here, this is one chromosome, each half is called a sister chromatid. So this is one chromosome, but this is also one chromosome, and it's confusing because, again, if I go back up to here, it looks like there's two separate chromosomes, but they're not really separate because they're attached to each other at the centromere. So one chromosome. Eventually, after replication, and this again is a little confusing the way your book is presenting it, this all happens in interphase, going from here to here. That happens in interphase. This condensing, that happens during mitosis. So this line is almost confusing. This happens in interphase during the S phase of interphase, and this happens during the first phase of mitosis. So before mitosis begins, we have to have them replicate. And this is where your book is, I'm not quite sure why it says it this way, but it's incorrect. I want to make clear that sister chromatids condense during mitosis, not before mitosis. So as mitosis begins, we get the spindle fibers forming, and those spindle fibers do attach to the centromeres. So let's go through and take a peek at what's going on. Again, with interphase, we have the cell is very normal. This parent cell is very normal. It's got the nuclear envelope, the nuclear membrane. It's got the duplicated chromosomes. You can see that centromere right there, so you know they're duplicated. So you know we must be in the G2 phase or after like the end of the S phase. So because we have our replicated chromosomes. And also we have these little structures here called centrioles. So interphase, we're just all ready for mitosis to begin. In the first stage of mitosis, this is called prophase. I've already told you pro means early or before, so this is the first phase, prophase. In prophase, 4 important things are happening. Number 1, the nuclear membrane starts to break down. You can see this little dotted line here that's breaking down. Number 2, the chromosomes that each have a sister chromatid, those start to condense or get shorter, long and spindly like chromatin, short like chromosomes. That happens. The next thing is that, and it's not listed here, but I like to list it, is the centrioles start to separate from each other. They're starting to move away from each other. At the same time that these centrioles are moving away from each other, the spindle fibers are forming, and your book does mention that one. And those spindle fibers are eventually going to attach to our chromosomes. So that's prophase. Those are the 4 things in prophase. The next is metaphase. Meta means middle, and these chromosomes are lining up in the middle. Notice that they're kind of lined up like this. If this is the head of the chromosome and this is the foot of the chromosome, they're kind of lined up head to foot. There's a little bit of overlap there, but they are still separate chromosomes. They're just lined up in the middle. Also notice like the centromeres kind of are in the center of that cell. And we call that very, very center part, we call that the equator, or we sometimes call it the metaphase plate. So that's what you see here. And notice that the spindle fibers are attached to the centromeres, and that's actually helping them line up in the center. It's arranging them kind of in that line on the center. The third phase is called anaphase, and ana is to separate. So in anaphase, we have the sister chromatids are actually splitting apart from each other. And one full set is going up to this, maybe we call this like the North Pole, right, of this centriole. And these chromosomes remember these are sister chromatids here, so this one's an exact copy of this one. We have this whole exact copy set going towards, I guess we could call that the South Pole. So these chromosomes are separate, the sister chromatids are separating. In telophase, 4 things happen. And it turns out that the 4 things that happen are kind of the opposite of prophase. And telo means end phase, so this is the end phase. Number 1, the nucleus starts to reform. Number 2, the spindle fibers disappear. There's no spindle fibers, nor are there any centrioles anymore. So that's 3 things. And then the 4th thing is that the chromosomes unwind. So they're unwinding, and they're starting to resemble the long uncoiled chromatin that we saw before. The second thing that your book mentions, and this is important, that telophase and cytokinesis happen at the same time. But cytokinesis is the pinching and the pinching in two, and it's not telophase. It's just that it's kind of happening at the same time. I like to use the analogy of it's like rubbing your belly and patting your head at the same time. They're two different actions, but they're happening at the same time. Or singing and dancing, right? Two different things that are happening exactly at the same time. So cytokinesis is where the cytoplasm divides. You can see that's starting to happen during telophase. So that's your basics of mitosis. I strongly urge you to watch the videos that I provided on Blackboard by the Amoeba Sisters or by Crash Course Science to give you even more details. These are the basic details that you need to be successful in the lab that's coming up. One of the things that we know about cell division is that when cell division is not controlled, remember the stops in the cell cycle that we talked about earlier? When cell cycle is not controlled, when it just keeps going and going and going through the cell cycle without even a breath in between, that's actually called cancer. So cancer is characterized by unrestrained cell growth, and of course this can cause serious health issues. Cancer is the #2 cause of death in the United States after heart disease. So this is a big deal. About 20% of all people who die in the United States are dying from cancer. So we've talked about what can cause mutations that can lead to cancer, like radiation or chemicals like cigarettes, those kinds of things. Or it can actually just be caused by the random mutations of the way that cells replicate themselves, the way DNA replicates itself. In any event, cancers definitely happen, and there are things that you need to know about. So let's get a bigger picture here on what cancer is. So there's some things that we know about cancer. It has 3 very distinct characteristics. And the first characteristic that I'm going to talk to you about is that cancer cells have something called, well, let me restart this. Normal cells have something called contact inhibition. Contact inhibition, there's your term right there, contact inhibition. Normal cells have contact inhibition. What that means is that when normal cells are in contact with each other, when they bump up against each other, they stop dividing. The cells in this petri dish will not divide because they're touching each other and they're normal cells. However, cancer cells have no contact inhibition. That means when they're bumping up against each other, they don't stop each other from dividing. They just keep dividing and dividing and dividing. And that's actually what's happening here, is that they're just piling on top of each other. And that's one of the 3 characteristics of cancer cells. They have no contact inhibition. The second one is that they divide indefinitely. Remember how I said about the telomeres earlier? That was like the first thing we talked about in this chapter, where telomeres are like the odometer, and they, if the telomeres are gone, then the cell kind of dies, apoptosis. So that's normal. We normally want cells to not keep dividing. But cancer cells do something interesting, and that is that they do, in fact, rebuild those telomeres. And because they rebuild the telomeres, they can keep dividing and dividing and dividing. Now, when they divide and divide and divide, they're doing it so fast that they're not spending a lot of time in G1. And remember, G1 has to do with growth. So cancer cells are actually a bit smaller than regular cells because they are just dividing so fast. The third characteristic of cancer cells is that they have something called reduced stickiness. Normal cells are absolutely going to be sticking to each other. I mean, you're a multicellular organism. I hope that your normal cells are sticking to each other. That's a good thing. With cancer cells, their stickiness is reduced, and therefore they can branch off. And when they branch off, that allows them to metastasize and move into your body, the rest of your body. So, hold on a second. Okay, this is a little bit better. The words aren't covered up by the picture. So again, I just talked to you about about the characteristics of cancer, and we talked about how this had no contact inhibition. Well, something that cells that don't have contact inhibition can be called tumors, but not all tumors are cancerous. So this is important to know. Benign tumors, benign means fine, it means good. Those are things that can be removed safely. They're just masses of normal cells that don't spread. And remember, for it to be cancer, they have to spread. So some examples of benign tumors might be a mole on your skin, something along those lines. Can remove safely, not invading other parts of the body, totally fine. A malignant tumor, mal means bad, so malignant tumors are actually considered to be cancer because they are spreading throughout the body, and anything that spreads throughout the body is called metastasis. So tumor, that's this no contact inhibition, but spreading through the body, that's this dividing indefinitely and having less stickiness. And so what you see here is a metastasis throughout the body. All of these regions here have tumors. And those tumors are dense because the cells are smaller, and so forth. So, so you've got all these tumors. And we know that cancer can kill you, but strangely and kind of surprisingly, it's not the cancer cells themselves are not toxic. There's nothing inherently bad about the tumor, but mostly, I'm sorry about the cells, but mostly it's because the tumor gets larger and larger, and as it gets larger and larger, it's taking up nutrients and it's taking up energy, and importantly, it's taking up more and more space. And as it's taking up more and more space, like here and here, as it's taking up more and more space, it's actually pressing up against the neighbor cells, and it can actually block other cells from carrying out their normal functions, and it can kill them because they're just taking up so much space. So that's what happens to people with cancer. It's like the tumors just start eating up all of the space and dividing and dividing and dividing, and that's what kills you. So how do you treat cancer? You are probably familiar with the idea of chemotherapy and radiation. Both of which are known to slow down the growth of these tumors. For chemotherapy specifically, you have drugs that are interfering with the way that cells divide. So any cell that's dividing rapidly is going to die. Just remember that we talked about how maybe the cells lining your intestines replace themselves fairly often, every 3 weeks or so. So people who are on chemotherapy, it's killing their cancer cells because those are dividing rapidly. But it's also affecting your intestinal cells or any other cell that divides rapidly. And so we have these unpleasant side effects from chemotherapy that include like the ability, you get tired, but you also can't really take in nutrients, you feel sick to your stomach, that kind of thing. Radiation also disrupts cell division, but instead of it circulating throughout your whole body like the chemotherapy drugs do, radiation is very pinpoint, and it's using this high energy radiation exactly where the tumor is located and kills those cells. Now, this isn't perfect. It can harm nearby tissue, and that's not good. But it's also really, because it's so pinpoint, it can be very effective. Again, what causes cancer? We've talked about this several times already. Chemicals, radiation, and normal natural things in cell division processes. Is there a cure for cancer? Right now, we don't have a complete cure for cancer, but there are lots of successful therapies and treatments. They are conducting research on how to maybe even look at the DNA that's controlling the cell cycle, that maybe that's how they're going to have future treatments and maybe cures for cancer. So that's all that I have for you on mitosis. The rest of this lecture and the next video, which will also be on this, we'll be talking about the sexual reproduction, and that's meiosis. And with meiosis, we really are focusing here on sexual reproduction, but we'll also talk about fertilization. We'll talk about what sex is, and how some species can use like both methods of reproduction. We'll just hit that very lightly. So sexual reproduction and meiosis. In order to understand sexual reproduction and meiosis, you need to understand a little bit more about chromosomes. And just kind of keep in mind that you are a whole being, right? But you got half of your information from your mom, and you got half of your information from your dad. And so you being this whole being, we use the term diploid. Diploid. Ploid refers to chromosome number. Di means having two. So literally, diploid organisms are organisms that have two copies of each chromosome. You are diploid, okay? Your parents are diploid. But how did you get information from both parents and you still have the same chromosome number as them? And what happens is that those chromosomes, when they get separated into sperm and eggs, they become something called haploid. And haploid means that they only have half the amount of chromosomes in there. So if we get like to humans, if we really just specifically talk about humans, we can say that an adult female has 23 pairs of chromosomes, and an adult male has 23 pairs of chromosomes. Each has a total of 46. 46 for female, 46 for male. When a female makes eggs, and when a male makes sperm, you cut the number of chromosomes in half. So instead of having 23 pairs, you just have 23 total. So instead of having 46 total, you have 23 total. That's 1/2. The only way that you as an individual could get back to having the normal chromosome number of 23 pairs or 46 total is through this process of fertilization. And fertilization, you have this fertilized egg. The fertilized egg has the proper number of chromosomes, and if that fertilized egg goes through the full developmental process and is born, you have this offspring that is fully complemented with its chromosomes. So fertilization is when two haploid cells merge to create the diploid individual. We know that individuals, they resemble their parents. We do know that, but they don't resemble their parents exactly, because you get half of your information from your mom and half of your information from your dad. So there's two big things that happen here in meiosis, and each of them happens in a different stage of meiosis. Number one, it reduces the amount of genetic material in the gametes. Remember, going from diploid to haploid. And number two, it produces gametes that differ from one another, with specifically about the types of alleles that they carry. Remember, we've used this word alleles before. Alleles are the different versions of the gene. And remember, your mom and dad would very likely carry different versions of the gene. So with meiosis, when you're making siblings, you don't give each sibling exactly the same combination. You can give them different combinations. And we'll talk about how that happens as we go through this process. And we'll learn about it a lot in lab. So we're going to make sperm and egg, right? And I told you that you have these pairs of chromosomes, and all of this is true. But I wanted to put this in context of sister chromatids, which we've talked about before. Sister chromatids are not the pair that I mentioned. No, in fact, this is one chromosome. Remember, this is one chromosome. So this is not the pair. Instead, what the pair refers to is something called homologous chromosomes, or shortened, instead of saying homologous chromosome, you might say homologs. So this is homologous to this. Basically, this chromosome that you got from your mom has a lot of the same information that the chromosome that you got from your dad has. You know, maybe the gene for eye color is on there. Well, it's also on there. And maybe the gene for, you know, height is, or one of the genes for height is there. Well, it's also there, right? These are very, very similar chromosomes. So chromosomes that are very similar in content, but maybe they differ in their alleles. That's called homologs. Those are called homologs or homologous pairs. So each of the pairs go through their own replication, making their own sister chromatids attached at their own centromere. All right, so kind of keep that in mind as we're going through. Meiosis has 2 stages, but of course you can't go through cell division unless you've gone through interphase first. So interphase, just like with mitosis, interphase is not part of mitosis. Interphase is not part of meiosis. It's the stuff that has to happen before meiosis or mitosis can happen. And really, it's the same thing. We're just making sister chromatids. So here we go, interphase, we're making sister chromatids. And just focusing on these 2 homologous pairs, this one from mom, this one from dad, we say that this is one homologous pair. Same thing here. This one's from mom, this one's from dad. This is one homologous pair, 2 chromosomes. During meiosis 1, homologous pairs separate. If you look in each of these cells, this chromosome only has one, this cell only has 1 chromosome. This cell only has 1 chromosome. We started out with 2 chromosomes, still 2 chromosomes. At the end of meiosis 1, we are now haploid. Diploid, haploid. So that happens. And then meiosis 2 has to happen because you can't have your sister chromatid around. Got to get rid of that. So we go through the process of getting rid of sister chromatids. These cells are also haploid, but they're haploid in the proper way because they don't have their sister chromatid. These are haploid as well. They just unfortunately still have their sister chromatid. So we got to get rid of that. Notice that there's 1 chromosome in each of these cells. So that's the heads up. That's the big overview. And now we're going to dive into looking at the details. Okay, so in the division 1 of meiosis, we have, before we can get to that, we've got to have our interphase, which means we're going to make our replicated chromosomes. We've got, oh look, there's a little centromere right there, so you know they're replicated. We have our centriole off to the side, we've got our nucleus and so forth. Okay, so we're going to enter meiosis now. And meiosis, something really weird happens. Homologous chromosomes line up right next to each other in this form that, as you'll learn in your lab manual, this form is called a tetrad. So we have these 2 homologous chromosomes hanging out in the tetrad. And you see that each pair of homologous chromosomes, in this case we have 1, 2, 3, 4, 5, 6. We have 6 total chromosomes, or 3 pairs. Each pair lines up with its own homolog and does this really strange thing called crossing over, where the maternal chromosome gives a little bit of itself over to the paternal, and the paternal chromosome gives a little bit of itself over to the maternal. So they're trading each other a little bit. You can kind of see that end result right here. They're still in the tetrad, but you can see they've traded little bits of the chromosome. Okay, so that's the special thing that's happening in prophase 1. Tetrads happen, crossing over happens. That's a big deal. But it's still prophase. And we learned that 4 things happen in prophase. The nuclear envelope dissolves. The chromosomes coil up into this chromosome shape instead of this chromatin shape. The centrioles start to separate from each other and move towards opposite sides of the cell, and the spindle fibers still happen. So those 4 things still happen, but we've got this extra thing here happening, tetrad and crossing over. In metaphase 1, we've got something different going on. We have the homologous chromosomes lining up at the equator, right? There's the equator, the center of the cell, North Pole, South Pole, equator, but they are lining up with their tetrad. So they don't separate their tetrads yet, they stay together. Remember, I'm just going to go back really quickly and look at mitosis and notice how that they lined up. They're not with their tetrad. If they were with the tetrad, this green one would be with this one, but that doesn't happen in mitosis. In meiosis, it absolutely does happen, and there you see it right there. So in metaphase 1, you line up with your homologue, and it's the homologue that goes in the center of the cell, in the metaphase plate or the equator. In anaphase 1, remember anaphase means to separate, and so what we have being pulled apart here are homologous chromosomes are being pulled apart, and they're moving to opposite poles. The sister chromatids are still attached to each other at the centromere. And of course, the sister chromatids aren't exactly the same anymore because they've got this like mix-up of maternal and paternal right there, and so forth on each of the chromosomes. After anaphase comes telophase, and of course cytokinesis happens at the same time as telophase. But let's take a look at what happens in telophase. We have 4 things happening. We have the nuclear envelope reforming, we have the spindle fibers disappearing, we have the, in this case, we have new centrioles forming, and we're going back into that spindly kind of chromatin shape. When now these cells are ready to, each of them, to go into prophase 2. And this is the second cell division. The good news about prophase 2 is if you can learn mitosis, you can learn meiosis 2. Because mitosis and meiosis 2 are basically exactly the same with one little exception. And that is that in meiosis 2, our cells are haploid already. And we're becoming more and more haploid. In that we're gonna go ahead and separate our sister chromatids apart. I guess you can't say you can become more haploid, but you can become appropriately haploid. That's a little bit better way of saying it. All right, so 4 things happen in prophase 2. The nuclear envelope disappears, the centrioles move to the poles, the spindle fibers form, and we go into the chromosome condensed shape. In metaphase 2 of meiosis 2, the chromosomes line up each in their own equator or metaphase plate. In anaphase 2, we separate those sister chromatids from each other. And in telophase, which we're lumping together with cytokinesis in this case, just to make life easier, the 4 things happen. The nuclear envelope reforms, the spindle fibers go away, the centrioles go away, and the chromosomes uncoil and become they become basically, in that chromatin shape. So you got 4 chromosomes. Sorry, you got 4 cells, each with their own unique set of chromosomes. All right, this seems like a pretty good place to stop. So I'm going to stop here. We're going to pick up in the third part of this when we get back together in just a minute.